Hello, everyone. Welcome to a new episode of the Dissenter. I'm your host, as always, Ricardo Lopes, and today I'm joined by Dr. Francesca Bellazzi. She is postdoctoral researcher in the ERC project Assembling Life at the Center for Philosophy and the Sciences in the Department of Philosophy, Classics, History, Art and Ideas at the University of Oslo. She is interested basically in the complexity of the interactions between different entities, and in particular, she is fascinated by the interface between chemistry and biology. And today we're going to talk a little bit about philosophy of science with a focus on biochemistry, and we're going to discuss what are functions, what are kinds, specifically biochemical functions and kinds, genes as well, and some other related topics. So Dr. Bellazzi, welcome to the show. It's a huge pleasure to have you on.
Thank you. Thank you for inviting me. It's a huge pleasure for me to be part of this show.
Thank you. So let's start perhaps with functions. I mean, I would like first to ask you more generally in the context of science and philosophy of science, what are functions?
Yes, well, that's an excellent question. And I would say that, like, in a nutshell, so if we try to bring together all the different accounts of functions, functions are special causal contributions, which can give us like informations about the identity, the history, and the context relativity of the role that an entity plays. So using a classic toy example, we can take the heart. The heart is a muscle that has the function to pump blood, or at least this is one of its functions. And the function of pumping blood is a special causal role that the heart plays in the physiological system. It tells us what makes the heart a heart compared to other muscles, because it specifies what the heart is doing, but it's also telling us that the heart has been selected as a muscle to pump blood. So as you can see, the notion of functionality can inform us on a lot of different things. Now, functions are also attributed to artifacts, for instance, and I think that the definition that I gave, that is that functions are really special causal contributions, can work for something like a chair. Like, we can use a chair to do different things, but what makes a chair is that it has the function of allowing us to sit on it. And this kind of like allows us to define the object. Having said that, so having provided this kind of general definition, I do think that when we apply functionality to different types or different kinds of objects, then we might need specific account of functions. So in that sense, I think that after the first step of providing like a general definition of functionality, we can be quite pluralist about specific notions of functionality for different objects. So I think that the way in which we ascribe functions to artifacts, such as a chair, might be different from the specific way in which we want to ascribe functions to something like the heart or any other trait, and that is different also from other kinds of entities like molecules.
Mm-hmm.
Yes.
Yeah, so when it comes to functions, it just came to my mind because, for example, when it comes to the heart and it pumping blood, I mean, in that specific case, and probably others in our human bodies, are we talking specifically about evolved functions? There is the functions that a particular organ, for example, a particular biochemical molecule have evolved to perform, or can it also include other types of, secondary acquired functions, for example?
Yeah, like this is an excellent question. I think that probably different philosophers would give different answers to this, and there is no like unified view. I would say that I am quite sympathetic with the idea that in order to identify what is special about functions as causal contributions, we need to bring in the evolutionary history. So like the function of the heart is what it has been selected for. Now, whether this just includes pumping blood, for instance, for the heart, or whether it can include also what are called like distal functions, I don't have a strong argument for neither of these views. I would say that generally I'm quite happy to ascribe functionality if we can identify a form of evolutionary background but this doesn't necessarily have to be like direct. And I think that this is how we can bring in, for instance, evolutionary talk into ascribing functionality to molecules, because molecules are not the classic target of evolution. They are a bit different. It's not like they present different features compared to, for instance, traits or phenotypic traits in particular. So yeah, in that, like compared to maybe other philosophers, I'm a bit more loose in the fact that what I think is interesting is a form of general evolutionary context, as that is helpful to identify like which of the causal contributions are functions and which are just causal contributions, so roles that the different traits can perform.
And when you talk about distal functions, which is something you mentioned there very briefly, for example, when it comes to the heart, since it pumps blood, would the distal function perhaps be that it also distributes oxygen through the body to the different kinds of cells? Would that count as a distal function?
Yeah, that would be a kind of like distal function that is quite proximal, as if we unpack like the pumping blood, then pumping blood is useful for for moving the oxygen. Another like distal function can be, for instance, the sound that the hearts make. So the fact that, like, for instance, this can tell us whether something is alive or not, or can be one of the parameters that can be used. And there philosophers differ in opinions about whether, for instance, the sound that the heart makes is a function or not, because somehow it's such an effect of what would be the, like some people would call the proper function, that maybe doesn't count as such, even if, for instance, it can have a role in the interactions that like an animal or even us can have with other living beings.
In that case, the sound would be like what, a byproduct of the heart pumping blood, and not exactly a function, right? I mean, it's not the function of the heart to produce sound. The sound is simply a byproduct of it, right?
Exactly. And then we can even say that like the heart hasn't been selected because it is an organ that makes a sound. Like the heart as a trait has been selected in a series of animals because it pumps blood, so it allows for circulation.
And we'll probably get into more of this when we talk about biochemistry specifically, but since I earlier asked you about evolved functions and also what I called, I'm not sure if this is the correct terminology, but I call them acquired functions. For example, we know that across different species, different animals, plants, etc., the same biochemical molecules can be present for different, or can have different functions in different contexts. So, I mean, what would count there as an evolved function? Would it be the original function of the molecule, and then when it acquires new functions in more recently evolved organisms, for example, then it would be an acquired function? Or, I mean, how do you deal with that?
Yeah, excellent. Well, I think that one of the core things about functionality is that, or at least in how I think about functionality, is that functionality is always context dependent. So we always need to consider the function of something in a given context. So let us take a concrete example that is one of those that I have studied myself, and we can take, for instance, the function of vitamin B12. Vitamin B12 as the function in humans, or as many functions in humans actually, but one of them is to contribute to erythropoiesis. So it intervenes in this process that regards the renovation of red blood cells. Now, this biochemical function is quite specific to the presence of erythropoiesis and to the fact that like erythropoiesis in humans has been selected in a way that thanks to which vitamin B12 can basically display its function. However, vitamin B12 is only synthesized by bacteria and archaea of specific species, and there it has a different function mostly an enzymatic function there as well, but of course it's not a function in erythropoiesis because bacteria and archaeas don't have blood nor red blood cells and so on. So in that case, we can notice that we have the same entity, which is the vitamin B12 in general in all of its vitamers, and these entities display different biochemical function. Why? Because they contribute to different biological processes, which have been selected to operate differently. So I would say that, for instance, it doesn't make much sense, or rather, more than doesn't making sense, it's not really informative, for instance, to say that vitamin B12 has a function in living beings in general, because we don't get like the specificity that we need. And while instead, if it is more meaningful, or it is more informative to say vitamin B12 has a function in bacteria and archaeas because it can act as a coenzyme in many different physiological processes of those species, and it has, for instance, a function in erythropoiesis in humans and other mammals. So we make it specific to the process that has evolved and in which the vitamin displays its role.
And then all of those different physiological processes it participates in across different organisms, are all of them are functions of that specific biomolecule, right?
Yes, in a way. Yes.
And so let me just ask you one more question before we get into kinds, because it just came to my mind when you mentioned the example of the heart pumping blood. So let's say that, for example, someone develops cancer. So when that happens, of course, the blood also reaches the cancer and sort of feeds it with nutrients, with oxygen and all of that. I mean, since it's sort of what we would consider an abnormal process to develop cancer, could we also say that in that specific case, one of the functions of the heart is to feed the cancer or not?
Yeah, excellent question. So I think that in this case, and this is why bringing in the evolutionary context is really important, in this case, we can say that the heart is contributing to a dysfunctioning process, which is the process of cancer replication from the perspective of the organism. And in that case, the heart is not doing what it has been selected to do, in a way that is like the heart hasn't been selected to feed cancerous cells, for instance, like by providing oxygen, but the heart has been selected to pump blood across the organism. And then somehow what is dysfunctioning in itself from the perspective of the organism, of course, is the cancer. And this is, and that's why I think that, bringing in evolution can really help, because we can say the same about, for instance, the role of vitamin B12 in erythropoiesis for forms of blood cancer, for instance. Like in that case, of course, there is still the causal contribution. It's not that vitamin B12 stops intervening. We don't have, unless there are, like, for instance, severe lacks of vitamin B12, but that's a bit of a different case. But what happens is that it is the process to which these molecules are contributing that is dysfunctioning. Why? Because it's not doing what it has been selected to do. Right. So it is behaving differently. And I think that this is really a powerful tool that bringing in evolutionary consideration to functionality F, because it can allow us to do these distinctions between these functions and functions.
Right. So let's get into kinds then. Of course, we've probably already mentioned some biochemical kinds here, but what are kinds more generally in science?
Yes, so this is, of course, like an excellent question, especially when we're talking about ascribing functions to different kinds. And I guess that we can take at least a dual answer to this, a kind of like minimal answer and a more elaborated answer. And I think that, like, the minimal answer is that, well, kinds are just types of things. So we can, like, use kind talk to just mean that, well, things are clustered into different categories, and we can distinguish them because they can be clustered into different categories. And this is a kind of like weak use of kind talk. I think that in philosophy of science and in metaphysics of science in particular, the term kind is a bit more loaded than type talk. And it generally refers to the fact that we tend to classify objects or processes into group of things, and these groups of things are kinds. Now, these kinds can at least be divided into being real or natural kinds. And here I'm conflating the distinction, even if, of course, there is a huge debate on whether we can do this. And more conventional or instrumental kinds. So we can classify things into clusters that we think correspond to real divisions in nature, or we can classify things into clusters which are just useful for scientific purposes. And then in that, we don't really have a commitment into trying to, for instance, understand whether there are real distinctions in nature. And I think that classic examples also here is that we can take different molecules of gold and classify them under the kind gold. And with that, we want to say, well, there is something, such as at least the gold molecules, which share something in nature for which we cluster them together. Or we can take, for instance, the category vegetables in nutrition, which seems to be much more of an instrumental kind. Why? Because we are clustering together different things because it is useful, but this distinction doesn't correspond to, for instance, a botanical distinction, as stuff like spinach or tomatoes would classify as vegetables in nutrition, but their biological classification is different, and technically tomatoes are a fruit, even if we wouldn't consider them as such, and related. So I think that this is quite an important contrasting category. And I do think that there are some kinds that fall in between. So now I'm also working on the notion of investigative kinds, so kinds that are good for investigation, in which we can use to try to unpack what's really going on. And I think that, for instance, the kind vitamin is there into the investigative notion, and the kind gene as well, which I know we can talk about later, is something that can go into that direction. That is, it has been introduced as conventional or as instrumental, so we don't want to have commitment in the actual world about the existence of something such as the gene, but the introduction of the term allowed us to discover so many things, to the point that now at least maybe some, I mean, I would say that, for instance, the molecular gene is a natural kind, so it corresponds to something existent.
Mm-hmm. Yeah, so we'll probably come back to the investigative kinds later when we talk about genes then, but let me ask you, when it comes to natural or real kinds, how do we determine that something is a natural kind? Are there specific criteria?
Yeah. So again, like extremely debated topic, and I would say that I wouldn't want to commit on a definition that has to be valid in all of these cases. I do find that, of course, we need to do then empirical investigations also to corroborate our theories. But generally, I need to say that I am quite fond of a theory of natural kinds that has been proposed by Mohamed Ali Khalidi. And I think that his account is quite powerful in that it at least gives us some tools to distinguish natural from conventional kinds. And these are the fact that these kinds present properties which are projectable. What does it mean? It means that we can do prediction and explanation based on inferring the presence of a property from one kind to another. So the moment in which we consider these properties that are associated to the kind, we are able to predict and explain stuff about instances of the kind. And the fact that these kinds have a role in our causal network. Now, this role is dual in the sense that the instances of the kinds have a causal impact on the world because of the properties they display, so they can enter into causal relation, and also the properties of the kinds are linked by causal connections. So I think that this tool, or this conceptual tool, is quite powerful to distinguish kinds, or rather conventional from natural kinds, or at least give us reason to investigate whether a kind is natural. So they can also be seen as a starting point for naturalness.
Mhm. Does what we consider natural kinds depend on the level of analysis we're using? So I was thinking that, for example, in biochemistry, we usually refer to molecules, but if we go down one level and we're looking at it through the lens of physics, would the kinds there be the elements or even the subatomic particles? How do we approach that?
Yeah. So I need to say that I am quite generous in my kind description. So I would say that there are all these types of kinds. I know that like other philosophers like Thomas Taco that you interviewed and others would have a much more sparse account of natural kinds. They would say no, like the only natural kinds are maybe like the physical subatomic particles. Or if one wants to take like a very bottom-up approach, you know, the only natural kind is the universe, and then everything else is just a component. While I would say that I'm personally quite generous, I think that yes, it depends on the type of entity that you are considering, and that kinds can really be clustered in a way that doesn't even have to be hierarchical. Like I'm quite happy with like cross-cutting kinds. Or kinds that are like in different, like the same entity can present different types of kinds as well. So as you said, like if we take even like complex molecule like water, then water can be a kind, and then we can unpack it and we can see that there is oxygen and hydrogen, and then we can unpack it and see that there are protons, neutrons, and electrons, and so on. And I personally don't have a problem with that. So my ontology is quite busy. There are a lot of things.
Yeah. No, I was just asking because, I mean, if we want to be as reductive as possible, there are people out there, philosophers of science, scientists that would probably argue that the only real natural kinds are the ones that we can find at the physics level. And so things like subatomic particles
Yes.
and stuff like that. And all the rest that we find above that level are not real kinds, I guess.
Yes. No, I don't like that. I think that the word, like, I need to say that as a kind of rule of thumb, I do find causality quite important. Like, if something is an important causal role, then we should take his ontological profile at least seriously. So we should at least investigate whether we have a kind there. So, yes.
And what to get into biochemistry specifically, what are biochemical kinds then?
Yes. So, well, again, here I start from a term that comes from the discipline biochemistry, but I think that in my understanding of biochemical kinds, we find all those molecules, macromolecules, compounds that have a role in living systems and that actually present a combination of structural, functional, and evolutionary properties. So this is how I understand them, because of course, like I take that from the term biochemistry, but I want to include also maybe kinds that are studied by structural biology, by molecular biology. So basically like all those chemical entities on the one hand that display also these properties that are relevant for life and for biological beings. So classic examples are proteins, which are of course one of the main cases, but I would also include vitamins, carbohydrates, lipids, which are crucial for membranes, and the DNA and RNA, which are crucial for heredity, and also protein synthesis. So these are all molecules that maybe like in the different departments are studied by different people, but I think that they share important properties. And my characterization of them is that linking it back to what we were saying about kinds is that in order to have these biochemical kinds as I intend them, we need to have some chemical structural properties that we need to identify. And the investigation in itself of the notion of structure in biochemistry is quite interesting because these molecules are macromolecules most of the times. And so they don't fit easily into the discussion of chemical structure in the philosophy of chemistry literature, because normally they focus on stuff like either single elements, which are the mostly discussed case study, or things like water, which in a way are is still much simpler than something like a protein, for instance. Then they need to have a function, and this relates to the whole discussion on like proteins functionality, but basically like is this role into biological processes. And this brings in evolutionary considerations. That is, these molecules, again, don't display random functions. They display functions in processes that have been selected. And they can have this causal contribution role. So, and maybe even, but this is really something that I'm still trying to work out, is that we can maybe even put a constraint on the fact that these molecules should be biosynthesized, or there should be the possibility of them being biosynthesized, that is synthesized by organisms, even if, of course, these now, especially with the new tools that have just been awarded by the Nobel Prize, for instance, in which we can predict new molecule structure, design new molecules for proteins, maybe this criteria is a bit too strict. But I think that still the fact that like these molecules need to play a role in processes that have been selected is important. So again, the context is important.
And so let me just ask you, when we're talking, for example, about proteins or about lipids or about DNA, for example, of course, they have smaller components. They have building blocks, like in the case of proteins, we have amino acids. In the case of lipids, we have fatty acids. In the case of DNA, we have nucleotides. So would those be kinds, or how would you deal with the building blocks there? What would you call them?
Yeah, I would say that they are definitely chemical kinds. So again, like given that I am quite generous in my kinds of description, I think that they are chemical kinds. We can identify chemical structural properties. Probably they can fit easily into forms of microstructuralism, which is generally the criteria applied for chemical kinds. So I think that, yes, like they would be so. I think that basically these components become biochemical in the moment in which they are assembled in the right way. So I think that that's important, and in the moment in which they can display the functionality that they have. So in the moment in which we can, think about the function that they display and the role that they play in organisms that have evolved.
That's a very interesting point that you just made there. So is it possible to identify exactly at what point a molecule becomes a biomolecule, or when it's just an inorganic molecule, let's say?
Yeah, I think that probably it's not easy to identify the exact moment, but I would say that the moment in which it plays a role in a biological system, it becomes one. So I do have this quite dynamic view of the interactions between the so-called chemical and biological interfaces, or chemical and biological domains. And I think that, if I would put it in a nutshell, in a kind of catchy way, which is not super precise, but maybe gives the idea, is that a chemical molecules become biomolecule, or a biochemical in the moment in which it plays a function in an evolved system. So there is this component. I know that there are counterexamples of like tricky cases. So for instance, what about magnesium or lithium? They play important physiological roles, while we would still understand them as mostly chemical entities. At the end, they are elements. And I think that there is like an interesting case in which we have a chemical element or a chemical kind that displays a biochemical function. Because in the moment in which magnesium is contributing to specific physiological processes, or even when they say that like magnesium aids sleep, this is a very like, kind of maybe a very distal function, because we're looking at a really an overall organism thing. But this is one of these cases in which we have chemical kinds, like elements that display a biochemical function. Why? Because they enter into selected processes and they manifest like specific causal contributions. I don't think that we can really be the kind of like police there and like saying, oh, you are biochemical, you're chemical, like all the time, because I think that a lot of analysis should be done case by case. But generally, there is this idea that in the moment in which we find an interaction in evolutionary processes, we find something biochemical. And if we want to make it even more strict, then we can say, well, not only that, but also the fact that the processes have been selected to interact with those molecules. So again, context.
So in that specific case, we would determine them as being biomolecules functionally and from an evolutionary perspective as well, right? I mean, because we can find, for example, lithium and other ions and other molecules also in geological processes, for example, in rocks and stuff like that. But in those specific cases, since they are not performing a biological or a physiological function, they are not biomolecules, even though they are exactly the same kinds of molecules, right?
Yes, exactly. This is exactly what I would say. Yes.
And so let me just ask you, because within the domain of proteins, we have a very specific subcategory that is enzymes. So how do we deal with them? How do you deal with them? Do you say that enzymes are also a kind or not?
Yes, I would say so. I would say that like enzymes are also a kind, and like in structural proteins are a different, or like molecular motors are different kinds. That is, these are like subcategories. And then again, given that I'm quite loose and I'm happy with having like cross-cutting kinds, I'm also happy for the same type of protein to display different roles even at that level. So one of my favorite examples are crystallines. Crystallines are moonlighting proteins, as they are called, exactly because they can display an enzymatic function in digestion and a structural function inside. And they do so basically by tweaking their tertiary structure a little bit. So per se, they are the same protein, like they're classified as the same protein mostly because they display the same primary and tertiary structure to a great extent, but there are differences, like slight differences in the tertiary structure, which allow the molecules to perform different roles, or simply basically the surface of the tertiary structure just gets slight modifications in a way that then they can display different functions, and by displaying different functions, we can classify them as like enzymes or as structural proteins in the different biological processes. So again, the fact that they happen to be in processes that have a different evolutionary history, like the digestive process and the side process, allows us to identify which properties are relevant.
Mm-hmm. But in biochemistry, since we are combining biology with chemistry, do you distinguish between biological and chemical functions? Is that something you do or not?
Well, so I think that, well, in biochemistry in itself as a discipline, like I'm not a biochemist, so probably my answer will just come from me studying biochemistry. But I don't think that there is a clear distinction between that. I think that the notion of function is used quite loosely and to mean different things in different contexts. I do think that as a philosopher, for instance, one of the useful things that we can do is to provide conceptual clarity by saying that there are different notions of functionality at play according to what we want to consider. So I think that it is important to distinguish chemical functions from biochemical functions, from biological functions, because these are going to tell us different information about the entity that we are considering. So chemical functions are quite loosely applied in description of functional groups. Functional groups are specific structural features of a chemical molecule or compound or element which allow them to enter into specific reactions. So it's basically what is telling us the reactivity profile of a molecule. As you can imagine, if we take the molecules that are normally considered in biochemistry, these molecules are massive. They are very, very big, and they will have loads of functional profiles. In principle, there will be loads of different reactions in which the molecules can enter. And this is why the moment in which, for instance, we want to consider the function of vitamin B12 in erythropoiesis or the function of hemoglobin in binding and releasing oxygen, we are not interested in all the functional profiles of the macromolecules. We are interested in very specific ones. We are interested in those parts of the molecules and those chemical physical properties, really, which allow the molecule to display the function in the given system. And I think that for simpler molecules, like for instance, vitamin B12, which at the end are forms of cobalamin, then we can identify some functional groups, which are the specific ones that are able to enter into the reactions relevant for erythropoiesis. But if we consider molecules like proteins, then not only we need to consider the chemical properties of the molecule, but we also need to consider the shape. So the tertiary structure is crucial because, again, here it's interesting why we need to distinguish chemical from biochemical functions, because, for instance, hemoglobin doesn't only need to bind oxygen. Hemoglobin needs to release oxygen at the right time. So we need to get this capacity to bind and release oxygen, for which the tertiary and then the quaternary structure is actually crucial. And then to this, we can add the biological functionality, which I think is really the selected effect view of functionality. I find is the most accepted and successful account of functionality. And that for that, we need evolutionary history. And I think that biological functionality can be applied differently to different molecules because they can display a more or less direct form of evolutionary history. So again, taking examples, vitamin B12 has a more direct evolutionary history in the bacteria and archaea that synthesize the molecule. So there we can identify what are the processes that have been selected to synthesize the molecule in the organism. While for the great majority of mammals, but also algae and other species which rely on vitamin B12, we need to consider a much bigger evolutionary history because we need to get vitamin B12 from those bacteria that are synthesizing the molecule. And this happens via a lot of different ecosystem interactions. And this is, of course, different from the evolutionary history of a protein that we synthesize, such as hemoglobin. Because in that case, we can identify, and other proteins, we can identify a specific set of genes or specific sequences which are encoding for the protein. And for some of these proteins, it's possible to have a direct evolutionary history. Like we can track it, we can compare it. There is a lot of really fascinating study on comparing the different evolutionary history of proteins. And there we can apply biological functionality in a different way because somehow it's more direct. So we can track it.
Right. One very interesting thing that you mentioned there or glossed over a little bit was the fact that at a certain point you mentioned the tertiary structure of proteins. And I mean, it's very interesting because at least in the particular case of proteins, if I understand it correctly, their function depends a lot on the way they are structured, right? I mean, their function sort of derives, at least to some extent, from the way they are structured physically, right?
Yes, exactly. And this is really fascinating because the folding can happen in many ways. So how we move from basically the result of the DNA expression to the tertiary structure, this can happen in many ways. But somehow what matters there is really the shape. Because basically it creates different surfaces that can then interact differently in different contexts. Which is why, again, there has been this the importance of, for instance, the discoveries awarded by the Nobel Prize are so important because they allow us to go from primary to tertiary structure, which is where the functionality happens.
Mm-hmm. And so let's talk then a little bit about genes. Earlier, at a certain point, I think, if I remember correctly, you talked about genes as a sort of investigative kind. So tell us about that. What is a gene exactly?
Yeah, so I think that genes are a very fascinating notion because they developed the kind of extension and intention of the gene term changed a lot through time. And I think that it is quite important to specify which type of gene we are talking about. So if we are talking about a molecular gene, or if we are talking about a Mendelian gene, and at which point in the history of molecular biology we are talking about genes, because the term has been really changing. Now, I would say that if we want to make everyone happy, a common understanding of the gene is the one of the molecular gene, and then the gene becomes a stretch or a string, often contiguous, of DNA and RNA that is encoding for the primary structure of a protein or other functional RNA molecules. And this is the kind of generally accepted definition. We also find the definition of the Mendelian gene, which are these groups of actually stretches of DNA which encode for a trait. And as you can imagine, the scale of this is quite different, because in the molecular gene definition, we are considering just the bits that are going to encode for a protein, which of course is quite long, but is still way less complex than, for instance, the gene for the nose or for the heart or for other traits. So I think that these are the two big notions that we find in the literature. I mostly worked on the notion of the molecular gene, as this is really where a lot of the interactions between chemistry and biology happen, and between life and chemical entities happen. And I think that the definition that I gave, while it is informative at a first stage, is actually quite inaccurate in the moment in which we consider new discoveries, in particular the discoveries of the last 40 years, and what is called like the post-genomic revolution or the post-genomic change, in which, thanks to the new discoveries that we have made, we notice that actually genes are not, for instance, identical to contiguous stretches of DNA. And to this discovery, and that actually, for instance, polypeptide chains can be transcribed by non-contiguous stretches of DNA, that the same stretch of DNA can encode for many different products. So there is a lot of complexity there. And some of the reactions to these have been eliminativist, at least in a kind of ontological sense. That is, well, there is nothing like the gene. The gene doesn't exist. We have DNA, we have expression, and this matters. I don't walk that route. I think that actually what we can do is that we can consider genes as temporal entities. So this is quite, I know, maybe a kind of crazy idea, but I think that it works because I think of genes as something that emerges or comes through expression. So they exist in the moment in which they are transcribed, basically. And if we take this temporal notion of gene and this kind of flexible notion of genes, then genes can become entities that exist as realized by specific sequences in a given moment in time, but in another moment in time, the same sequences can realize or can constitute actually a different gene, which has a different function because it encodes for a different protein or molecular products.
That's all very interesting. And please correct me if I'm wrong, but if I understand it correctly, we don't have the entirety of the information we need to produce a protein in just the gene alone, because in going from the gene through transcription to mRNA and then translation through tRNA and then getting the protein itself, we go through several steps where post-transcription factors, for example, play a role, right? I mean, so what I'm trying to say is that when it comes to the information necessary to produce a protein, it's not entirely in the gene itself, right?
Yeah, this is very true. And again, I think that the context in which gene expression happen matters massively. Like, I really love this definition from Griffiths and Stotts, Carola Stotts, that genes are things that cells do with their genome. Because this gives the idea of the fact that the cellular context is massively important for the determination of all of this behavior and all of this reading. And then, of course, with that, all the factors around the sequences, which you mentioned, again, recently awarded discovery of macroRNA and the role that it has in these processes, the fact that genes are regulated, the fact that there are networks all of this matters massively. So like genes are highly contextual entities in the sense that they are really realized by this very complex machinery, which is offered firstly by the cell, and then even by the whole organism matters. And I think that this probably was one of the most surprising things about some of the research projects, for instance, the Human Genome Project, is that they were hoping that in the moment in which you get the sequence, you will get all the information that you need. And this, of course, and this has proven to be quite disappointing, because actually it is the way in which you read the sequence, the way in which the sequence is transcribed, the whole interactions that happen in the cell, that for a lot of phenomena are really important. This, of course, doesn't mean that there aren't specific cases in which actually having the sequences is telling you everything you need. Like, luckily for medicine, we know that there are some diseases that are caused by very specific sequences, and the fact that we have this knowledge allows us to intervene. But generally, for the great majority of the other genes and for a lot of complex phenomena, the interaction is way more complicated than just the realization of a sequence.
Mm-hmm. Yeah, it's very interesting. So there's a lot, if I understand it correctly, there's a lot of context dependency here when it comes to genes and the link between, for example, genes and traits or genes and proteins, right?
Yes, exactly. I do think that there is, that context dependence is really crucial for all of the biochemical stuff, really. So genes in particular are an important case, but also for everything we talked about, I think that context is crucial. Now, some philosophers I know take context as a threat to naturalness or as a threat to reality. Like if something is context dependent, it's like less existent. I don't endorse that. I think that first, it is important to distinguish context dependency from discipline dependency. That is, okay. For instance, like it's not the fact that I am a biologist or a chemist that matters for my understanding for the gene. It can influence my empirical study, so I can decide to focus more on some properties rather than others. But this is really like an epistemic distinction, a domain distinction, and this is really epistemic. And maybe that can actually matter, for instance, for naturalness or non-naturalness or kindhood. But I think that context dependence is something different, as this relates to the actual interactions that happen in the systems that we are considering. And I don't consider that as a particular threat, also because I think that everything is actually really context dependent, because every time we don't consider context dependency, we are making an abstraction. So we are getting rid of parts of the context in which something exists, which can be really useful for some specific purposes. But then I actually find that, yeah, things really get to be what they are because of the context they are in and the relationships they are in matter for their identity. And I think the genes are a paradigmatic case, but also what we talked about earlier, like lithium in a mountain or magnesium in a mountain is going to be something different from me taking my magnesium before going to sleep because I want to sleep better. Let us say.
Yeah, no, it's really very interesting. And I was thinking when you mentioned that there are people out there who, whenever we talk about context or context dependency, they think that things who are context dependent are less real than things who are not context dependent. Do you think that could be because many philosophers of science and even more so scientists themselves have this sort of substance ontology, and I mean, if they adopted, for example, a process ontology, then it would be easier for them to understand processes themselves as being real instead of substances, for example.
Yeah, I think that there is definitely something like that. I don't think that substance ontology necessarily is against context, but I find that definitely the focus on intrinsic properties is. So I think that there has been historically this preference for intrinsic properties. Like I think that if one studies the history of philosophy, of Western philosophy, in the great majority of cases, this has been the case. And of course, there is a value to it. Why? Because intrinsic properties favor stability. Stability is stability, so it's easier to identify, it's easier to rely on. So there is this, and it is quite useful. I do find that it's not that the process of abstraction in which we focus on what is stable are necessarily self-defeating for empirical or epistemic purposes. I just think that in the moment in which we want to bring back the entity into reality and we want to get an overall characterization of what something is, then we need to be open to consider relational properties as well and extrinsic properties. And then definitely something like a process ontology or even forms of ontic structural realism can be really insightful in our understanding. So I don't think that there is necessarily the need to like get rid of objects or get rid of substances, but I think that we need to become a bit looser in the type of properties that we think are relevant for these entities, especially when we consider entities that are really at the borders, like biochemical entities or genes, for which really the interactions matter.
Right. But because genes are so context dependent, and we also have or add post-genomic discoveries, like, for example, the fact that the expression of genes is regulated, and so we have epigenetics, and then also, as we mentioned earlier, we have post-transcription factors playing a role in the kinds of proteins that are produced, etc. Can we still consider genes natural kinds or not?
Yeah, excellent question. I do think that like molecular genes can still be considered natural kinds, but we need to consider the relevant properties for it. So we need to consider the fact that genes are not simply structural entities, so they're not simply specific strands of DNA or RNA for some species. The fact that genes needs to have a functional profile is very important, and this brings in also the evolutionary history. So those three properties that I mentioned earlier about biochemical kinds, so the fact that biochemical kinds need to have a structure and they need to have a function and they need to have an evolutionary history, matters for our consideration of the naturalness of the genes. I do find that like genes are natural because they can be quite projectable. So the moment in which we consider genes in terms of their expression, we are able to predict and explain a lot of things, which is simpler for cases, as I mentioned earlier, in which the stretch of DNA that realizes the gene is quite stable. But this is still possible for like a lot of research. And also genes display a core role in our causal structure in the cells because, well, they express the protein. So, and they encode the protein, which and other very important molecules, and this has a crucial causal role.
So there's one last topic I would like to ask you about, and I think it's completely unrelated to what we've been talking about up till now, but I want to ask you because I found it really interesting. You've written on the application of virtue ethics to the context of the COVID-19 pandemic. Could you tell us about that?
Yes, yes. So this is, really a side project. I need to say that I think of myself as like a philosopher that does mostly philosophy of science, but I am interested in a lot of philosophical questions. So I have a lot of, kind of like side projects and side interests. And I guess that those papers, as for many of us, really came from the fact that we got hit by this like very harsh reality. And especially for myself, I was stuck in far away from home. I am originally from Italy. And as we know, like the north of Italy in particular was really impacted by the pandemic. And a part of me felt that I needed to do something about it, which I think was what a lot of people felt. And my idea was like, okay, like there is a lot of talk which seems to be based on ethical schemes that, again, are quite rigid. And they don't consider the fact that like a lot of our actions are done by individuals in context. So I feel that probably there is this social context which comes in again. And so I wanted to suggest a paper in which, if we bring in virtue ethics, which not only focuses on the actions of the character of the individual, but also has this flexibility regarding what each individual can do in each context, compared to, for instance, a more deontic ethics or a more utilitarian ethics or consequentialist ethics, which seems to be at a much more like a group level or either like at a principle level. I thought that, exploring that would provide a different angle to a lot of the talk that, especially in the UK, which is not a surprise, but there was a lot of like utilitarian mindset. Like we need to think about what is good for the majority, at least at the beginning, because then of course the situation changed. But there was a lot of like, okay, we need to think about what is good for the majority, what is going to maximize the outcome. And then if something is going to get loose or someone is going to get loose on the way, whatever kind of thing. And that really surprised me in a way, or rather hit me personally. And I was like, okay, we need to think about these things from a different angle. I also found that those type of talk in that context was also taking away agency from all of us, because again, deciding for the majority is not something that an individual can do unless you are a politician or someone that is in the ruler position. While instead deciding every day what I can do to contribute to my situation, which is what virtue ethics invite you to do, gives us more agency, more control, even in situations of little control, but you're not anymore a spectator of choices, but it can also be quite empowering. So I think that those were some of the motivations.
But in this particular case of the COVID-19 pandemic, which virtues would you say are the most relevant to consider here?
Yes, that's a good question. I do find that generally like the virtue of prudence as it is understood, which is basically trying to evaluate in each circumstance which virtues you should use, was probably the most relevant. Prudence is also the most difficult virtue because of course it requires a lot of analysis and control. I also find that forms of bravery, not in the sense of bravery as, oh, I go outside and face the danger and I fight the virus, because unfortunately we can't do that, but bravery in the sense of, okay, I might sacrifice some parts of my well-being if this is going to help someone else, if this is going to help someone that is maybe more vulnerable than myself. So there is this element of like, okay, maybe wearing a mask is not pleasant, but if wearing a mask can save someone or can at least contribute to preventing that someone that is in a really vulnerable position gets infected, then I can choose as an individual to do so. So probably like prudence, benevolence, courage, like being brave were definitely relevant virtues.
What about humility? Do you think that it would also make sense as a virtue in the context where perhaps we have to rely a lot on what experts are saying?
Yeah, that's a very good point. So I think that humility play a role, but I understand, I think that we shouldn't go with the kind of like pop understanding of humility in that case. I think we should really like consider the definition of intellectual humility that is provided in a lot of the literature on the topic, which is the fact that we need to be able to evaluate when is the moment to step back and when is the moment to come forward. So when is the moment in which I don't have the skills to, for instance, understand the technicality of a problem, but I do have the skills to understand maybe the social component of it. And I actually think that probably the lack of good intellectual humility has also been problematic on all sides, because, for instance, the fact that we were following maybe only the information that were coming from the sciences, which is very important because they tell us the mechanism thanks to which the virus replicates and spreads. They weren't though informed by, for instance, studies in sociology, psychology, okay, how do we deal with loneliness? And somehow the social sciences got pushed back a lot. And that was also a case in which somehow there wasn't much intellectual humility, for instance, at the public, at the policy-making level, because some sciences were really, really put very high and others were put very low and not considered. And probably both at the individual level, in which I can bring in my skills And maybe, I don't know, if I am a teacher, I might need to learn from scientists how the virus spread, but then as a teacher, I might know what is good for my students and the fact that maybe they need specific interactions. And so we need to find ways to give students those interactions, and also the policy-making level, so bringing in a form of like good intellectual humility, in which different sciences and different perspectives come in on different domains.
Mm-hmm. Yeah, because mentioning humility, perhaps there was also not much humility, at least on the part of certain types of experts, like, for example, doctors, epidemiologists, and so on, immunologists as well, because when it comes to, as you said there, the social aspects of how we dealt with the COVID-19 pandemic, things like loneliness, isolation, and stuff like that, maybe people who could have played an important role there, like psychologists, sociologists, were pushed aside, and the stage was given more to, as I said, doctors, epidemiologists, immunologists, and people like that, right?
Yeah, no, exactly, which I think that's something else that I don't think is underlined enough, is that like these situations are always complex, and so they might require complex solutions. So the fact that we bring in psychologists and sociologists, for instance, on trying to understand the impact that loneliness can have, especially on vulnerable categories like kids or teenagers, but really on everyone, doesn't imply that then the fact that it is still true that the virus spreads in many ways is pushed away. Like the ideal thing is really to find an interaction between the two, which is of course super difficult, and in a situation of emergency that we faced, and especially the Western world hasn't faced anything like that in the longest period of time. So of course it makes sense that we didn't have the tools to see that clearly, but somehow the contrast wasn't really, to put it simply, like, oh, let us lock everybody up, or let us just let the virus spread because people need sociality. Like, probably the ideal solution was trying to find a common ground between the two. And in this, I do find that probably spreading education and empowering the individuals in making choices could have been really useful, because I also feel that giving this stage to just the experts that were talking mostly in experts term just made people feel extraordinarily even more frustrated and isolated, because they didn't have the tools to understand what was going on. And that was a problem not of the people that didn't know or didn't understand. That's a problem of those that communicate such knowledge. And that's, again, something else in which, for instance, the role of sociologists and psychologists and maybe even philosophers for us could have been helpful, because I think that people should have the tools to understand stuff that matters to their life. So to this, like, I do have a project with a colleague of mine, Simone Picenian, like the right to science and the right to have access to science, and why it is important to remove epistemic barriers. And it is particularly important for contexts like the COVID pandemic, vaccination, but even climate change, in which people are required to take action, and the action is action that they need to take, but on the basis of scientific knowledge that no one is really explaining them. And this is really not empowering, and we should actually, flip that and, and explain people what's going on so that they can become even better agents. And maybe in this I am naive, but I do believe in the power of education.
So with that in mind, with your belief in the power of education, do you think that this kind of work and people reflecting a little bit on what individuals themselves can do and how to develop perhaps certain kinds of virtues, do you think that in a possible future pandemic, this kind of thing could be taught to people, and to, I mean, even in schools and other places like that, and to help people, children, adults, deal with that, with what's happening in such a kind of context?
Yeah, I do think so. And I think that this is really something that all of us that work in education, that have an interest in science communication, in knowledge communication, via all sorts of tools like podcast interviews, public speech, public writing. I think that this is really something that we have to work on, and that we need to communicate. I think this is really important. And unfortunately, the fact that there is a lot of focus on, I mean, of course, we need the technical research as well to investigate the work, but I do think that it is important to bring this knowledge everywhere, because I do think that this can make us better agents, or at least can have an impact in making us better agents, or giving at least the capacity to choose in a more informed way which type of agents we want to be and how we want to interact.
Great. So just before we go, would you like to tell people where they can find you and your work on the internet?
Yes. Well, I do have a website, which is just my name, francescabellazzi.com. And I keep my website up to date so that you can find what I'm doing, where I'm going, which talks I do. And there is also a section on public outreach, which is called Articles and Media. And there you can find my pop articles. I try also to be quite active philosophically on Twitter, or now X, which, with the handle Franci Bellazzi. So you can also find what I'm doing and which talks I'm going to, my conferences, and everything that is philosophy related there. So thank you.
Great. So I'm leaving some links to that in the description of the interview. And Dr. Bellazzi, thank you so much for the great conversation. It's been really informative and fun. So thank you.
Thank you. Thank you. Really for me as well. Really a pleasure and an honor. And thanks to everyone that has been listening to this.
Hi guys, thank you for watching this interview until the end. If you liked it, please share it, leave a like and hit the subscription button. This show is brought to you by Enlights, learning and development done differently. Check their website at enlights.com, and also please consider supporting the show on Patreon or PayPal. I would also like to give a huge thank you to my main patrons and PayPal supporters: Per Ergo Larsen, Jerry Mullerens, Frederick Sunda, Bernardo Seixas, Olaf Alex, Adam Kessel, Matthew Whittingbird, Arno Wolf, Tim Hollis, Eric Alenia, John Connors, Philip Forrest Connolly, Dan Demetri, Robert Windiger, Rui Inácio, Zup, Marco Neves, Colin Holbrook, Phil Kavanagh, Mikkel Stormir, Samuel André, Francis Fortiago Nunes, Fergal Kossen, Hal Herzog, Nuno Machado, Jonathan Labyrinth, João Linhares, Stantantia, Samuel Correia, Eric Hines, Mark Smith, João Eira, Tom Hummel, Sardas France, David Sloan Wilson, Yacila Dez Araújo, Romain Roach, Diego Londonho Correia, Yannick Puntar, Adana Rusmani, Charlotte Bliss, Nicole Barbaro, Adam Hunt, Pavlo Stasevski, Nelek Baka, Madison, Gary G. Alman, Sam Afzal, Adrian Yeager, Paul Tolentino, João Barbosa, Julian Price, Edward Hall, Edin Bronner, Douglas Fry, Franca Bortolotti, Gabriel Ponce Cortez, Ursula Litsky, Scott, Zachary Fish, Tim Duffy, Sonny Smith, John Wiseman, Daniel Friedman, William Buckner, Paul George Arnaud, Luke Loaki, Georgios Theophanes, Chris Williamson, Peter Wolozin, David Williams, Diogo Costa, Anton Erickson, Charles Murray, Alex Shaw, Maury Martinez, Coralie Chevalier, Bangalore Atheists, Larry Dealey Jr., Old Erringbon, Sterry, Michael Bailey, Dan Sperber, Robert Grassi, Igor N., Jeff McMahon, Jake Zool, Barnabas Radix, Mark Campbell, Thomas Dovner, Luke Nissen, Chris Story, Kimberly Johnson, Benjamin Galbert, Jessica Nowicki, Linda Brandon, Nicholas Carlson, Ismail Benzliman, George Coriatis, Valentin Steinman, Per Crowley, Kate Van Goller, Alexander Hubbard, Liam Dunaway, BR, Masood Ali Mohammadi, Perpendicular, Jonas Hertner, Ursula Goodenough, Gregory Hastings, David Pinsoff, Sean Nelson, Mike Lavigne, and Dios Necht. A special thanks to my producers: Isar Webb, Jim Frank, Lucas Stafiniak, Tom Vanagdam, Bernard Ugni, Curtis Dixon, Benedict Mueller, Thomas Trumbull, Catherine and Patrick Tobin, Giancarlo Montenegro, Alnick Ortiz, and Nick Golden, and to my executive producers Matthew Lavender, Sergio Quadriano, Bogdan Kanivets, and Rosie. Thank you for all.